Okay, so this morning I said I'd go a little bit into the cholesterol controversy, and it's always popular because we've heard about little else for 50 years. And this part 2 will put cholesterol into context. Now, I'm going to talk about cholesterol particle number, like your LDL is your low density lipoprotein, you've got trillions of them, but when you count them, it's seen to be a much better risk measure than just getting the cholesterol content, which is the old LDL bad cholesterol. So I am talking about the particle number, but most of what I say, or all of what I say, will still apply to trying to use the old LDL as well. But this is just the new super LDL. So I mentioned there the Semaine people, Catavans the same, they have nearly no heart disease, they're incredible, even though they're different, diverse genetic peoples, they have no heart disease. And I suggested these were some of the key reasons they had none. The low insulin, low glucose, no diabetic physiology, no MET sin, hyperinsulinemia sin, no hypertension. Hypertension does not increase with age with these people, unlike everyone in the Western world. No central obesity, obviously, and omega-3, omega-6 ratio. Now, they have other benefits from their environment as well, like they've got culture, community, they're out in the air and in the sun all the time working, but there's all that stuff. But the interesting thing is, these guys and these guys have the same or higher average particle count as these guys here in the West, who are in a mess, right? The highest CVD in the world. So it's a comparative analysis, we call it. Sure, the particle count can still be a cause and a driver, but there's some serious exceptions to it. If you've got people with no heart disease who have higher numbers than the people who are riddled. So that's just the point to be made there. And again, we're going to target these set of good things, and we're going to see these come up again in future slides. Now, another illustration, and I got slides or studies like these 6 years ago when I started this, which intrigued me in my early days. And you can see here the ApoB tertile. Now, ApoB is LDLP, it's the particle number, essentially. And you can see here that for people who have a high total cholesterol to HDL ratio, I mentioned earlier, that means they're probably insulin resistant, they're not so healthy. Well, for the middle and high ones in this study, sure enough, with higher particle counts, there's higher hazard for cardiac events. But something you must note is that for the people with low cholesterol ratios, who I would say are insulin sensitive and relatively healthy arteries, the higher particle count, if anything, means lower heart disease in the future, right? So this is a dichotomy, this is a paradox, but it's not really a paradox, because the people who are genuinely healthy with no inflammatory issues, the particle count doesn't really matter. That's the suggestion. So you got to know which person you are. Am I a person with a high particle count where it will encourage me getting heart disease, or am I a person where it's moot? It's not really relevant. My vasculature is healthy and these particles just float around benignly. That's a big question. So I gave the cholesterol conundrum back in 2014 and explained all of these lipoproteins, LDL, HDL, how they work, their residence time, how they interacted with the body, and a lot of stuff around disease too. But that was the cholesterol conundrum. It's on YouTube. But a few months ago I decided I'm going to dig into the particle count thing because people are talking about it more and more. As the old LDL-C begins to fall away and people are realizing it's kind of silly to use that for risk, LDLP is being talked about. So here's my office and studying away there. But I had to extend to my podcast table because I had nearly 200 papers and I print them all out because I like to underline, cross-reference. Working from a screen is hard when you get into this level of depth. So a lot of work went into it, and this is what I got. No, I'm not going to, this is the process of atherosclerosis from the LDL particles getting into your artery wall across the endothelium to the massive inflammatory cascade that can result. But we're not going to go through all of this science. It's fascinating, it's complex, but it's well established and there aren't too many controversies with that cascade. We're going to focus rather on the LDL hypothesis, and this is the kernel of it. If you have more LDL particles in your blood, more will get into your wall and you will have issues. That's it. That is the LDL hypothesis. The old one was LDL-C, the new one new and improved, is exactly that. And it's quite simple. It's very simple. You've got more LDLs, more go in, boom, you get heart disease. Very simple. Don't need to be a scientist. However, there are layers here of evolutionary technology that will manage whether or not, or to what extent, the LDLs will get in the wall. So they'd be really important, but the LDL guys usually don't talk about these. They just talk about the LDLs go in, boom, right? That's a problem. 4 layers actually that I've dug up, and we'll talk through each one. Now just to orient you, here's your artery, a wall on each side, and here's the blood flow, the lumen in the middle, some red blood cells there, and here's all your happy little LDL particles, right? Blood flows along, wall on each side. And we're going to be focusing in here, just where the blood meets the artery wall, and here's where our 4 layers are that decide whether your LDL particle count will be a problem. So blown up a little, here's level 1, the Glycocalyx, and this is only discovered 30 years ago. It's kind of a forest of hair-like protrusions that come out of the inside of your artery wall, the inside surface. They're amino acids and glucose, kind of proteoglycans, and as you can see there, they have a sieving effect, and they manage LDL particles' access to the wall. So the Glycocalyx is part of whether or not you'll have a problem. Second is the endothelial, the single cell layer that forms the inside edge of your artery wall. And this is a single cell layer, and particles can come across the cells through a managed process, and they can leak between the cells. So here's another couple of ways that the particles can get into the wall. And we'll go through that. And then you've got your proteoglycans, structures inside the artery wall, and they can trap LDL particles, whereupon they become oxidized, they're broken up. And if you have too many of them getting trapped, you can build up cholesterol into foam cells and plaques. So whether or not these things grab a lot of particles also decides whether your LDL particles are a problem. As if that wasn't enough, there's a 4th hugely important one, and that's HDL efflux. So your HDL lipoproteins go into the wall, they pick up any problematic cholesterol that gets left over in here, and they magically take it back out again into the circulation and back to the liver. So it is magic. And they don't talk about it much, the drug guys, because the drugs to help HDL failed. Therefore, HDL is persona non grata, pretty much. You'll hear a lot of people in reports, oh, high HDL doesn't necessarily help you, and some people with high HDL have disease. And you'll hear all this anti-HDL rhetoric. And the reason is the drugs didn't work, so it's just a competitive threat, basically. So anyway, there's the 4 layers. 1, 2, 3, 4. Evolutionary wonder. We want to know what governs, determines, mediates, or influences this 3-layer leakage and the efflux down here. Because if you don't know what's going on here, you're almost wasting your time looking at the numbers, because that's the least of it. Extra numbers up here, more blah, blah, blah. That's the least of it. It is what's happening in this system. So first, the Glycocalyx, and here it is, an electron microscope image. Very, very narrow. It's a couple of 100 nanometers. There is a barrier layer that goes beyond it, but it's a very tiny structure of hairs. And here's a paper I dug up, 2008. Arterial glycocalyx dysfunction is the first step in the atherothrombotic process. The first step. That's interesting, because I always thought it was the particles go into your wall and that's the first step. Well, no. Interesting paper. See the result of that. So looking a little closer, here's the hairs of the glycocalyx. The LDLs tend to be managed in some manner they don't fully understand to limit access to the endothelial cell. That's in good shape. And here's one that's in a mess, right? This guy's smoking maybe, drinking Coca-Cola. His glycocalyx has gone to hell altogether. And here is where leakage and also the recruitment of inflammatory molecules, monocytes, and all kinds of issues begin to happen on that inner wall when you destroy your glycocalyx. Here's one team that did a study on it, and they said, well, let's take in a Petri dish endothelial cells all together and try and damage this structure and see how much more LDL internalizes into the cells. If we damage it, more LDL should get in there, right? And they found that it did. When they damaged it with neuraminidase, you had nearly 10 times as much LDL went into the cell. So that appears to be what happens when you screw your glycocalyx. They used cationized ferritin, which they knew was much stronger damage agent, and they verified that this structure had gone down to 5% or less of its original. So they damaged the hell out of it. And they got 20 times more LDL to get internalized in the cells. Now, this is not in a human, but it's illustrating a point of principle. In general, the papers for damaging the glycocalyx that I've pulled out, high sugar, SAD diets, hypertension, and disruption to your metabolism relating to hypertension, oxidative stress, oxidized LDL, but not native LDL, undamaged, interestingly. Smoking blows the hell out of your glycocalyx. And arterial morphology, the areas in our arteries that get atherosclerosis are usually at branch points, specific points. It's not all over. And those points, there's recent papers showing where you can lose your glycocalyx around 50% of its thickness. So they're beginning to tie that to glycocalyx, the focal nature of atherosclerosis. And ischemia reperfusion. When you do operations, they often see with new arteries put in place, you get rapid atherosclerosis. And they're beginning to tie that to the damage to the glycocalyx that occurs when you do the operation. It kind of falls apart. And we'll tie back now to our target list of good things. If you have these good things in great shape, like the catavans, right, you will tend not to have any of these things that will destroy the glycocalyx. So it makes sense. So these are things to target. Layer 2, the endothelium. We're in layer 2. So here is an endothelial cell, and they're all lined up on the inside of your artery. And transcytosis is the process by which LDL particles are managed through the cell and popped out on the far side complete. Endocytosis, they get broken up for the cell to use what's in them. Transcytosis, they get brought in by various receptors to transfer across the cell. So this is a design process. This is not an accident. You'll notice the question marks because the latest 2018 paper I got on this by top people in the field, they admit that it's amazing we don't understand this process because they've done it for the blood-brain barrier and many other epithelial barriers. And they're just saying, wow, it's bizarre we haven't worked this out yet. One of the reasons is I don't think too many people are interested in this process because the simple theory of the LDLs jumping in and exploding is quite lucrative. Anyway, that's my thought. So this group showed it happening. They put gold particles, and you can see little gold black dots there, onto LDLs, kind of nanoparticles, so they could track them in a scanner through live cells. And they put the culture in, and after minutes, you had LDL particles being seen inside the endothelial cell, right? So they're coming in there. And after a matter of hours or less than an hour, they began to see them on the other side of the cell, effectively in the artery wall. They'd come right across and popped out the far side. And with the gold, they were a bit like kind of little Irish leprechauns with their pots of gold, taking them into your artery wall, or so to speak. So anyway, so this has been demonstrated. Now, they also saw that, I think this was in 12 hours or so, 25% almost had made the journey fully across. Now, evolution designed this process not to give you atherosclerosis, designed for other things. We can debate about that. Interestingly, another team looked at transcytosis, and when they put up the concentration of LDL by 10 times up here in the blood side, they only got doubling of transcytosis. So putting up your number of particles actually has a very muted effect on how many go across. However, another team used TNF-alpha, and this is an inflammatory agent. When you're inflamed, obese, diabetic, all this stuff, TNF-alpha goes up. It's part of an immune response. And when they brought in TNF-alpha, they got 4.5 times more to go across. So there's lots of things that can drive this process, and inflammation is one of them. Your brain, this is a little extra one. Does this happen for LDL to go into your brain? Well, I recently heard on a podcast that LDLs do not go across the blood-brain barrier. But there's a few papers on this, and here's one of them. They got the cells, and they had astrocytes or brain cells on one side, and they had a blood-brain barrier, and they saw that there was indeed LDL transferring across the blood-brain barrier. And they even tied down the mechanism. It was using the LDL receptor. They knocked out the LDL receptor, it stopped happening. They put back in the LDL receptor, it happened again. They did around 6 vectors of proof to say, you were happy. It comes across the blood-brain barrier in small amounts. They even got the idea, what if we take the astrocytes, the brain cells, and we deplete them of cholesterol? We take out some of their cholesterol. I wonder, will this speed up? And it did. Because when they reran the experiment, a lot more LDL was demanded and the receptors were upregulated to bring in more LDLs. So that's just interesting. LDL goes into your brain, and evolution is not an idiot. This was a designed process, right? So we'll go back to the artery though. The papers indicating what drives this process harder for more LDL to come in: C-reactive protein, inflammatory agent; oxidized LDL, oxidant induction, radical oxygen species. We know how to get those, right? Eat the wrong thing. TNF-alpha again, and lipopolysaccharides. So if you get infections or you have leaky gut and bacterial components come across your gut, their lipopolysaccharide is what activates your immune system. And in experiments, they've shown lipopolysaccharide introduced increases the rate of transcytosis. So lots of things that will drive up this transcytosis. Funny enough, they're the things you'll get if you break the rules on our target list, right? No doubt about it, nearly every one of them. So this is an important target list. One more, sorry, on the endothelium. Paracellular is the leakage of LDL between the junctions of the endothelial cells. And this happens too. It kind of leaks LDL in. And you've got 3 layers of tight junction technology there to manage this, but they can get broken down badly if you do the wrong thing. Here again, TNF-alpha introduced 4.5 times as much going through this route, right, past your endothelial cells into an artery wall situation. They even worked out that it was due to the apoptosis rate, or the cell death, that was induced by the TNF-alpha. And as the endothelial cells were dying and turning over, you were getting a lot more LDL through. And they got a great relationship there. The LDL permeability, straight line with the apoptosis rate, directly driven up by TNF-alpha. The other papers which indicate this gets increased, paracellular transport, TNF I mentioned, angiotensin II, which is related to high blood pressure, interleukin 17, that's immune response issues, or any insult to endothelial cells in your body, of which there are many, can drive this process. Go figure. This kind of stuff, right? This kind of stuff. Not exclusively, there are other inflammatory drivers, lupus, rare diseases, many other things besides this, but this is the big one for the modern population and the most addressable. Layer 3, the Intima Proteoglycans. These are funny creatures. They're down in the artery wall. They're strands of sugar and amino acids, and they do have a function to grab lipoproteins that are down here. And that's okay, that's part of their function. So the LDL hypothesis says they jump across, but we know now there's a lot more going on here than jumping across, right? But when they get in here, they can get stuck. But what we'd be interested in is what causes increased LDL reactivity and binding with these proteoglycans? Because LDL can go in and come back out again, but what causes them or drives them to bind? The papers I have that indicate what causes them to bind much more aggressively are blood from heart attack patients, corrected for cholesterol, same amount of cholesterol as a healthy guy, but the nature of their cholesterol. Blood from insulin-resistant people, blood from type 2 diabetics, all cause higher binding down here. Small, dense and oxidized LDL, yep, for sure. Higher ApoC3 content, which is associated with insulin resistance. You get C3 on your LDLs to greater level. Much higher affinity of this type of LDL. I'm just thinking back to Gary's talk of subclasses. Much higher when you have the bad subclasses. Type 1 diabetic mouse experiments. I'm not crazy about mouse experiments, but this one was a great one, very recent. They took type 1 diabetic mice versus wild type control mice. They were matched for cholesterol, and they said, why did type 1 get much more atherosclerosis? And they were a team that were focused in here. So they found out that the type 1 diabetic blood had 8 times more LDL retention than the wild type healthy mouse, just based on their blood and their LDL, right? So they don't just jump across and get stuck. There's a lot of things that decide how much they get stuck, right? That we don't get told about. We just get told about the number. So we're back again, yet again, sorry to be redundant, but the same kind of stuff is mediating and influencing this piece of the process. Layer 4, final layer, HDL efflux issues. So basically, when they do get down here, and they're going to get down here to a greater or lesser extent based on what I told you, HDL can go in through ABCA1 transporter. This is all understood and known, and HDL can take out the cholesterol from foam cells that has ended up here and can reverse macrophage and can fix the problem if you don't overload it or you don't damage your HDL. That's obviously a dependency, but it can. So let's look at this. This paper was superb, 2014, I think. A macrophage-specific cholesterol efflux capacity has been directly and causally linked to the prevention of atherosclerosis in animal models. So they wanted to go in and look in humans, what can if you have good HDL that can efflux very well, or you have bad HDL that's poor at effluxing, not just more or less now, but good or bad, what can it do? So they looked here and they saw having high HDL gives you a reduction in risk. And one of the reasons is because it loosely indicates, if you have higher HDL, that your system's in good shape and that your HDLs are healthy and can efflux, and many other things. So high HDL is a great indicator, but it's not infallible. Now, when they looked at the HDL efflux capacity, and the clever guys got the people's HDL, and for each person they tested with macrophage in a petri dish. They got all the different people's HDL and they fluorescent tracked to see how much cholesterol could it extract from the macrophage. This is quite amazing. And they measured it and they gave it a score for how healthy and how effluxy was each person's HDL. So that was an impressive 56% lower future heart attack based on the efflux capacity. Then they corrected this for the other risk markers that the people had. And they found out when you correct for the other markers, like I said this morning, dependent, independent, and try and mathematically correct, it got bigger. Nearly a 70% association, 70% lower hazard for heart attacks based on HDL efflux. That's a huge, if you had a drug that got a 20% reduction, right, it's a blockbuster. Interestingly, they had strokes here as well, and the corrected HDL efflux, a 90% reduction in future strokes associated with good efflux capability, which makes complete sense. I was just surprised at how big it was, right? So HDL getting your problematic lipoproteins when they do get in, getting them back out, is not a trick, it's part of the process. And if you screw that part of the process, you will have trouble. HDL is massively important, in spite of what the idiots say on the airwaves. These kinds of things, yet again, will generally overwhelmingly tend towards higher HDL, healthier HDL, everything else related to HDL. And that's why we always look at this list, because we're engineers and we focus on the big things that are most important. We don't get tied up in knots over some number, independent. So 1, 2, 3, 4 layers, right? Beautiful evolutionary complexity, all designed to work together to have your LDL sitting up here and be happy, even if you have a higher or lower number of them. And no one discusses them. We've shown how, generally speaking, our key list of reasons we would say the Catavans and the Semaine have got higher particle counts and no issues, lowest heart disease in the world, is they're fantastic on all of these and more. I have a bonus layer, and this is heresy. So you have to all agree to be partake in a heresy. A lot of lipidologists would not like the next few slides. Okay, you were warned. So here's a paper 2009, and it shows oxidized LDL going across into the LOX1 receptor, foam cells, atherosclerosis, blah, blah, blah. And LOX1 receptor actually is involved in every part of the atherosclerotic process. It's a receptor for mildly oxidized LDL. But you notice a problem. They're showing ox-LDL as being the problem in atherosclerosis, the mildly oxidized LDL in the blood. They're letting native undamaged LDL off the hook. That's no bueno. The lipid hypothesis is native undamaged healthy LDL is the problem. But this team were saying, no. It's when it's oxidized in the blood, mildly. Then it ends up going in, getting caught in the LOX receptor. So that's heresy, right? That a lipidologist would maybe not burn you for that, but certainly slap you, at least. So anyway, here's a team that tried to look into it, and I have a few of these papers who approached in different angles, got the same result. They got healthy controls on hypercholesterolemic patients. Now, these people have bad ratios, super high LDL, and they were patients. So in a way, they were kind of just people who had issues. You could view this. They found when they looked at the oxidation status of the LDL particles in their blood, not now gone into the wall, but in their blood, they found that they both had plenty of L1, L2, and L3, and these are not oxidized at all. These are native healthy LDL. But they found that the unhealthy people were the only ones who had L4 and L5, and this is very mildly oxidized, and this is called minimally oxidized, right? So wow, okay, so there's a distinction between the healthy and the unhealthy. So they got endothelial cells in a preparation and said, well, what are these particles going to do to endothelial cells? Remember, that's your barrier. You don't want to hurt your endothelium. And they found that even from the sick people or from the healthy people, L1, L2, L3, non-oxidized, cause no endothelial cell death. And even when they mixed them all together into a potion, L1 to L3 in a bag, and dunked it in there, didn't do anything. L4 and L5, different matter. 10% up to 40% endothelial cell death because oxidized LDL kicks off an inflammatory cascade, causes problems. They then got copper and oxidized the hell out of LDL, like it would be in your artery wall when it gets really oxidized, just to see, well, does that just blow away the endothelial cells? But interestingly, it wasn't much worse in endothelial cell death than the mildly or moderately oxidized. So these things in your blood, the team concluded, are an enormous part of atherosclerosis in your blood before they get into the wall. So they are conflicting with the LDL hypothesis. That is, healthy LDL get into your wall, then get oxidized, then you have a problem. They're saying, no, you got a problem in your blood. And the ox-LDL that measures these, there's an assay in the States, there's pretty huge hazard ratios for being high in ox-LDL as well, which does tend to suggest that this team have something to talk about. So the heresy could be true, but if that heresy was true, everyone would be looked at to see, do they have damaged LDL in their blood? So 1, 2, 3, 4, and bonus, layer 0, which if that was in any way true, even these wouldn't be needed as much. 5 layers essentially of what makes your LDL particle number be a problem or maybe not be a problem at all. If you stay on target, you target all the things that manage this whole 5-layer list, right? And then you might not need to worry at all about the number, be it 800 or 1500, you could say. And I'll finish with just one little illustration. You got an unhealthy high LDLP guy. I put 25 in there, but let's say that's high. And he's got some oxidized LDL and he's drinking Coca-Cola, or he's got an inflammatory condition. He's an unhealthy person. Through all these layers, I have no doubt he will end up with an inappropriate amount of LDL particles trapped in his wall. And his HDL will be in crap as well, so he's not going to be getting them out as fast as they're coming in. He's a guy who can benefit from medication, in fairness. Now, ideally he'll do lifestyle nutrition, but this guy could benefit from medication and trying to lower the number. But what about this guy, who's the same number of LDLP, but he's healthy, and all these layers are working fine, and his HDL is working great? He might end up with vastly lower problem in his artery wall. But if the doc's looking at the two of them, they're going to be the same high number, and they're both going to get the meds. If you're a high number, that's independent risk, it's causal. But none of them are really looking at the difference between these two people. So, most of it's in the book, if you want to get the book. But that is LDL Cholesterol Conundrum Part 2. And I'll have a Part 3 as well in a couple of months. Does that follow?